Hello everyone, my name is Julien Maruotti. I'm the CSO of Phenocell, a French biotech in south of France. So today I'm going to introduce you our latest model, which is an in vitro model used for acne research and active ingredient testing or discovery. As you may know, sebocytes are a key component of the sebaceous gland, which is involved in sebum secretion to the skin surface. The sebaceous gland also play a key role in acne development, which is a disease affecting up to 90% of teenagers worldwide. Multiple factors are involved in acne development, including key ones such as sebogenesis and inflammation. So Phenocell, using arachidonic acid stimulation and stem cell derived sebocytes, has created a new in vitro model which resume hallmark of acne development such as increased sebogenesis and inflammation. And I'm now going to introduce you the model and the data. So first of all, what are iPS? So iPS are pluripotent stem cells derived from blood or skin biopsies of individuals. So once we have blood samples from a donor, we will use forced expression of key genes to reprogram them into stem cells. These stem cells grow indefinitely and they can be differentiated into any cell type of the human body. So at Phenocell, we focus on skin cells, which means using iPS cells, we can produce unlimited amount of sebocytes, melanocytes, and keratinocytes, always coming from the same donor if one chooses to do so. In addition to the cells, Phenocell also offer ingredient testing services as a CRO and can perform on-demand and custom R&D. The advantages of using iPS technology is that first, unlimited amount of skin cells can be obtained from a single donor, and also several donors can be used to validate an ingredient and identify an ingredient which might be specific to a given population. So in this poster presentation, we use 3 donors, Caucasian, Asian, and African, to validate our in vitro model. The in vitro model was based on sebocytes obtained from iPS. The sebocytes were matured for 3 days in a culture medium. Then they were treated with arachidonic acid for 24 hours for the sebogenesis assay. And for inflammation assay, we use a treatment of 36 hours of arachidonic acid. So after thawing, sebocytes will display heterogeneous morphology with forming an epithelial monolayer. They will express keratin 7 and MUC1 in about 85 to 90% of the cells. Once the cells reach confluence, they are treated with arachidonic acid or with arachidonic acid plus a reference compound, which is isotretinoin in this case. So as you can see on this panel, arachidonic acid treatment strongly increases lipid production here. And the addition of isotretinoin has a significant effect on limiting sebogenesis, which was quantified in this panel here, where you see significant increase of lipid production following arachidonic acid treatment and significant decrease when isotretinoin is added to the arachidonic acid. So this means we can use iPS-derived sebocyte to model sebogenesis in response to arachidonic acid treatment. Next, we looked at the inflammation following arachidonic acid treatment. So the first thing you can notice is we did a panel of cytokine markers by qPCR, and for all 3 phototypes, you do see a significant increase in key inflammatory markers such as IL-6, IL-8, and TNF-alpha. And the increase is variable according to the phototype chosen, but is always significant. Similarly, treatment with isotretinoin significantly limits the inflammatory response in all phenotypes, but differences appear between phenotypes. For example, if you look at the African phototype and IL-8 response, you can see that isotretinoin treatment increased inflammation. So this shows that using iPS sebocyte from different donors, you can also reproduce a biological variability across a range of donors and check whether active compound has efficacy in all three phototypes or is more active in a given phototype. After doing the panel of inflammatory cytokine by PCR, we validated by ELISA and found that the results obtained by qPCR were also reproduced at the protein level with upregulation of the secretion of IL-8 and a decrease following isotretinoin treatment, which was in this case not significant. So as a conclusion of this research project, we have created an in vitro model using iPS-derived sebocytes and arachidonic acid induction that in a single model show both sebogenesis and inflammation, and those consequently can reproduce those hallmarks of acne. This model for sebogenesis is available in high throughput format and can be used to screen many compounds if requested. Thank you for your attention and for listening to me.